Bismillahirrahmanirrahim. I am Dr. Sana Jabin, and today I am here with the 6th lecture of our course Mycology and Plant Pathology, and this lecture is about Myxomycota. In our course, in the fungal systematics part, previously we have studied about the classification of fungi in detail. According to our syllabus outline, now we will be studying about the somatic structures, that is the vegetative mycelium of the members in Myxomycota, and we will consider a representative species from this phylum to study its life cycle and reproduction. This is the list of fungal phyla mentioned in our syllabus, which we will go through. Myxomycota, Oomycota, Chytridomycota, Zygomycota, Ascomycota, Basidiomycota, and Deuteromycetes as written in our syllabus, which is a class of the Deuteromycota. In some fungal phyla, the specific orders are mentioned, like among Oomycota, the order Pyronosporales, and among the Zygomycota, order Mucorales, Basidiomycota, Ascomycota, Erysipelas, and Physarum, Basidiomycota, Agaricales, Polyporales, Uridinales, and Astilaginales are given. We will be taking at least one species as example from each order to study its somatic structures and life cycle. Since there is no any order mentioned in our syllabus regarding Myxomycota, we will be taking an example from order Physarum, that is phylum, that is the order in the Myxomycota, and we will study the example of the Physarum polycephalum. Before starting a detailed account on this species, first we will start with the introduction. The term Myxomycota was first described by John Link in 1883, and this Greek word represents the slime molds. The myxo means the slime, and suffix mycetes represent the class in the fungi. Because of the superficial similarity of the fructifications of the slime molds with the fruit bodies of certain fungi, notably Gasteromycetes, although they lack any true relationship with these Eumycota, so they are included in the kingdom fungi, and the name is given Myxomycetes because of the fruiting bodies which resembles with the fruiting bodies of the Gasteromycetes. These organisms are commonly called the slime molds. Here in this picture, you can see a number of different species of the slime molds. These are present in any moist environment, and they can be found on the soil, and they can be found on the rotting wood, and they are very brightly colored. For example, here you can see the yellow color, the orange red color, the orange color, and other are mostly yellow in color. So these are commonly called the slime molds because of their slimy appearance. Slime molds have been studied mainly by mycologists rather than protozoologists. This is probably because they occur in the same habitats as fungi and are routinely encountered during fungal forays. Since slime molds are only rarely covered by zoology courses even today, so they are described here in mycological study, referring to the more specialized literature as appropriate. If you search about the classification of this group on internet, you will find variable classification schemes. To understand the precise identity of this group, we have to go to some authentic sources regarding the classification of this group. Here in this picture, you can see the classification of this group under kingdom fungi here, and this classification is not appropriate. I told you the details in the lecture 3 that was about the major fungal phyla. Index Fungorum is the most authentic community resource currently coordinated and supported by several well-known organizations of the world working on the field of mycology. If you search for a species in Myxomycota, for example, Physarum polycephalum, then you can see its taxonomic status here. It is characterized under kingdom Protozoa, phylum Mycetozoa, and class Myxogastria. So here it means it is not classified under the kingdom fungi, rather it is classified under the kingdom Protozoa. So this is its correct classification scheme. So here is a magnifying view of the classification of the Myxomycota. Its kingdom is the uh, Protista in some literature, and according to Ruggero et al., its kingdom is Protozoa. The phylum Mycetozoa, that is equivalent to the Myxomycota. Class Myxogastria, order Physareales, family Physaraceae, and we will studying a genus and species. The name is Physarum polycephalum, and this word represents it is author citation. Slime molds differ substantially from the Eumycota not only in phylogenetic terms but also regarding their physiology and ecology. Their vegetative state is that of individual amoebae in the cellular slime molds, or of a multinuclear or cynocytic plasmodium in the plasmodial slime molds. Motile stages bearing usually two anterior whiplash type of flagella may be present in the plasmodial slime molds. Amoebae or plasmodia feed by ingestion, that is the phagocytosis of bacteria, yeast cells, or other amoebae. This is followed by intracellular digestion in vacuoles. The mode of nutrition in the slime mold is therefore fundamentally different from the extracellular digestion and absorption as shown by Eumycota. So they differs from the true fungi, that is the Eumycota, and they are placed in a separate group is known as the slime molds, and it is not included in the kingdom fungi. Its status on the basis of DNA and its interpretations. Numerous phylogenetic analyses of DNA sequences encoding ribosomal RNA molecules and various structural proteins or enzymes have been carried out, but the results obtained are difficult to interpret because the comparison of different genes have led to rather variable phylogenetic schemes. Of the four groups treated in the Webster, it seems that the dictylostereomycetes, protostereomycetes, and myxomycetes are related to each other, whereas the acrasiomycetes have a different evolutionary origin. In general, the evolutionary background is, however, still diffuse in these lower eukaryotes. Now we will study in detail each class. First, we will consider the acrasiomycetes. They are also called acracid cellular slime molds or acrasia. The acrasiomycetes or acrasia, as they are called in zoological classification schemes, here you can see zool, it represents the zoological nomenclature or zoological classification scheme, are a small group currently comprising 12 species in 6 genera. Although appearing somewhat removed from the bulk of the slime molds, they still clearly belong to protozoa. The trophic stage consists of amoebae, which are morphologically distinct from those of the dictylosteroid cellular slime molds. We will study about these slime molds in detail in the next part. The amoebae of this group are cylindrical rather than flattened, and the granules behind the pseudopodium appears clear. You can see in this picture here These are the cellular slime molds. The arrow shows the direction of the movement of these slime molds. They represent the amoeba-like cells or called the amoebae. A represents the Limex type amoeba in acracid slime molds. You can see the absence of granules in the lobose pseudopodia. These pseudopodia are lobe-like, and there is a hair pointed structure which is knob-like, and it is also a characteristic feature of this group. In comparison with the amoeba of the protostelium with phylose pseudopodia. Phylose means pointed ended pseudopodia. Here these you can see the pointed ends, and these are the lobed pseudopodia, and its structure is slightly bulbous, making it a slightly cylindrical appearance, and this is slightly flattened. The posterior end is knob shaped and is called the uroid. Here you can see the knob shaped structure. Such amoebae are of the Limex type because their movement resembles that of slugs of the genus Limex. So you can see the slugs, they belong to kingdom Animalia, and the amoebae of these slime molds look like these slugs. So they are known as the Limex type amoebae because of the presence of knob-shaped structures or knob at the end. This is the uroid end. These slime molds are common on decaying plant matter, in soil, on dung, that is the animal waste, and on rotting mushrooms as well. But they are rarely recorded because of their small size, but can be observed using a dissecting microscope by taking a soil sample from their habitat. The most readily recognized species is Acrasis rosea, which has orange to pink colored amoebae due to the presence of carotenoid pigments in their body. The uninucleate amoebae feed on yeast cells, bacteria, or fungal spores. They can exist under unfavorable conditions, especially in the drought conditions, to form microcysts. These are the parts that help them in survival in the unfavorable conditions. Each microcyst germinates again to release a single amoeba. Sexual reproduction in the acracid slime molds is unknown. Next is the class Dictyosteliomycetes, also called dictyostelid slime molds. They live as both cellular and multicellular, unicellular and multicellular organisms. The Dictyosteliomycetes, in zoological nomenclature, they are known as Dictyostelia, is a group of cellular slime molds comprising 46 species in 4 genera. The best known example is Dictyostelium, which has been so named because the stalk of its multicellular sporocarp appears as a network made up from the cellulose walls secreted by the amoebae from which it is formed. Dictyostelium species are common in soil, on decaying plant material, and on dung, and can be cultured by smearing on non-nutrient agar with cells of suitable bacterial food such as E. coli, and adding a small crumb of moistened soil on the center of the bacterial smear. Amoebae will creep out of the soil and consume the bacteria. At the end of the feeding phase, the spore-bearing structures develop and can be isolated from this culture. Here in this picture, you can see the picture of the dictyostelid slime molds. Here you can see the sporangia. Here you can see the small unicellular structures. Amoebae of the dictyostelids are morphologically different from those of acracids in that they have phylose, that is the acutely pointed rather than lobose pseudopodia, as I have shown you in the previous slide. The next class is protosteliomycetes, also called the protostelid or plasmodial slime molds. In zoological nomenclature, they can be called protostelia. Protostelids are ubiquitous on decaying plant parts in soil and humus, as well as on dung or fresh water. They occur in all climatic zones from the tundra to the tropical rainforests. They produce amoebae with phyllosaudopodia. They feed phagocytotically on bacteria, yeast cells, or spores of fungi. Some species also produce small plasmodia, thereby providing structural affinities to both the cellular and plasmodial slime molds. Here in this picture, you can see the formation of sporogenesis hyphae in the protostelid. The A represents the early or pre-spore stage. B represents the head-shaped stage. Here you can see the head-shaped stage. C represents the appearance of the stereoregion. Here is the stereoregion. D represents the beginning of the stalk formation. E represents the later stage in stalk development with stereogene extended into upper part of the stalk tube. Here, F represents the mature sporocarp showing terminal spore with subtending apophysis. Here, and there is an outer sheet and the inner stalk tube present here. This picture is taken from the Webster and Weber book, that is Introduction to Fungi. Next in this picture, you can see an example of the protostilling slime molds. The A represents the fruiting sporocarp bearing stalked spores. Here, B portion of the surface of the sporocarp showing spores and their attachment. Here you can see the spores and their attachment. C represent the spores. D represent the naked protoplast emerging from the spore at germination. Here you can see this is the spore wall and this is the germinating spore. E represents the naked protoplast before cleavage, that is before germination, start of germination. Here you can see the cleavage and 4 cells are formed, 4 protoplasts are formed. Here a clump of 8 protoplast is present in the G, and the H represents the uniflagellate and biflagellate swarmers that is released from the 8 protoplast, and this G is converted into these biflagellated swarmers or uniflagellated swarmers. I represents the combination of these two opposite strains, and J represents the new young plasmodium. The next class is the myxomycetes. It is also called as the true slime molds or plasmodial slime molds. In zoological nomenclature, they are represented by the word myxogastria. They are the largest group of slime molds comprising some nearly 800 species belonging to 62 genera. These are the familiar slime molds so common on moist, decaying wood and other organic substratum. They are also abundant in soil. The vegetative phase is free-living plasmodium, which is a multinucleate wall-less mass of protoplasm. This may or may not be covered by the slimy sheath. These plasmodia vary in size and can be loosely grouped into 3 categories, particularly on the basis of their size. So these are the types of plasmodia based on the differences in their size. The first one is the protoplasmodia. These are microscopic structures which are usually giving rise to only a single sporangium. They resemble the simple plasmodia of protostelids. The second one is aphanoplasmodia. The aphanous means invisible. They are small, invisible, and thin open networks of plasmodial strands. Most aphaenoplasmodia are only seen with the help of dissecting microscope. Next is the phaneroplasmodia. The phanero means visible. They are large sheets of networks with conspicuous veins within which the protoplasm shows rhythmic and reversible streaming due to interactions of the calcium ions with cytoskeleton elements lining in the veins of this plasmodia. These movements can be observed with the help of a dissecting microscope. Here in this picture, you can see the phaneroplasmodia of Physarum polycephalum. A represents the margins of extending plasmodium. The protoplasm is particularly dense at the advancing edge here. Further behind protoplasm is a concentrated large veins. Here you can see the large veins which show rhythmic pulsion. B is the fusion between compatible plasmodia. Note here the complete fusion of the veins. C represent the lethal reaction following fusion between the incompatible plasmodia. This picture shows the slime modes that are the true slime modes, and according to International Code for Botanical Nomenclature, they belong to the class Myxomycetes, but in actual nomenclature, this is the class Myxogastria in zoological nomenclature. Now let's move towards the life cycle of a member of Myxomycota. I must say the member of Myxogastria. We will discuss the life cycle of Physarum polycephalum, which is a typical slime mold. The plasmodium is diploid and feeds by phagocytosis of bacteria, yeasts, or fungal mycelia, as well as fungal spores. It gives rise to a sporophore under appropriate conditions. The haploid spores are dispersed by wind or insects. These sporophore produce sporangia, and sporangia produce some spores which by the meiosis, and these spores are dispersed by wind or insects. In the presence of moisture, they germinate by releasing either amoebae or zoospores. Here you can see the spore germination. This is the spore wall and this is the protoplasm. They can either form the amoeba-like cells. These are called the Myxomoebae. You can say them the Myxomoebae because to differentiate them from the other cellular slime modes which have different function in their life cycle. Sometimes the spore germinate and produce flagellated cells which are called the swarmers. Myxomoebae are capable of asexual reproduction by simple mitotic division. Swarmers cannot divide but can readily and irreversibly convert into Myxomoebae. Under adverse conditions, Myxomoebae secrete a wall to form microcyst. So when the conditions are unfavorable, a wall is formed around the amoeba, and this is the inactive stage which is known as the microcyst. It is formed under unfavorable conditions. Both swarmers and Myxomoebae form phylous pseudopodia. Phylous mean pointed pseudopodia. You can see here the pointed pseudopodia of the swarmers and Myxomoebae, with which they can engulf their food. Sexual reproduction is initiated when two haploid Myxomoebae or swarmers of compatible mating type fuse to form a zygote, from which the diploid plasmodium develops. After the zygote formation, the phaneroplasmodium is formed. The plasmodium can survive adverse conditions by turning into a resistant sclerosium. So sometimes sclerotia are formed. These are the resistant structures which are formed after the unfavorable conditions in the diploid states. In these sclerotia, certain compartments are formed which are called the spherules, each containing several nuclei. Upon resumption of growth, or when the favorable conditions are back, the protoplast emerge from their spherules and re-establish the plasmodium, which is called the phaneroplasmodium in terms of myxogastria. Plasmodia can be maintained for prolonged periods in vegetative state, and sclerotia can be stored dry for months. Spores can be viable up to 50 years or more. The spores of Physarum polycephalum have been revived after more than 50 years storage in a herbarium. So this is all about the life cycle of the Physarum polycephalum, in which here you can see some notable things. Here this black color represent different type of nucleus, and this white represent the different genetic mode, and these two are, if compatible, they combine in the form of, here you can see the swarmer combined to form zygote, and Myxomycota can also combine to form the zygote. So this zygote can be formed from two different Myxomycota as well as from two different genetically compatible type of swarmers, and then they convert into phaneroplasmodium. And if the conditions are unfavorable, this phaneroplasmodium can form sclerotia, which help them to survive under unfavorable conditions. If the conditions are favorable, the sporophores are formed. These are the branches that bear the sporangia, and in sporangia meiosis takes place and spores are produced, which are haploid, and these haploid then resume their diploid state after the formation of zygote. So this is all about the life cycle of Physarum polycephalum. Now let's have a look over some pictures, original pictures taken from the internet of this organism. Here you can see the Phaneroplasmodia Physarum polycephalum growing on the wood chunk. And in this picture, Physarum polycephalum is grown on a petri dish. Here in the this is also an image of the Physarum polycephalum. Here you can see the veins, and here the veins form a network, and so many nuclei are present in these veins, and there is no septa, and this is the Phaneroplasmodia Physarum polycephalum. Here in the reference you can see that slime mold sings song. I want to elaborate it here as interesting information. You are hearing in this clip is the sound as a result of rhythmic movements of Physarum polycephalum in a petri plate. A little machine called the Energy Bending Lab was able to capture the electric activity of the mold and translate it into sound. This instrument is actually a series of modular tools that amplifies the mold's micro voltages and turns them into sound patterns called non-human rhythms. You can read the details about this from the given link. This is all about Myxomycota. For detailed study, please refer to the book Introduction to Fungi by Webster and Weber. Most of the content of this lecture is taken from this book from chapter 2, from page number 40 to page number 47. For other details, the link or reference of each source is given on the respective slides. Many thanks.